I'm Lara Szewczak. I'm a scientific editor at Cell, and I'm here today with Iain Cheeseman, a researcher at MIT. Whitehead.
Both.
Yes. And what I'd like to talk about today is what you study, first and foremost. And you've worked a lot on the Kinetic, which is this massive assembly, dynamic assembly of proteins that help anchor chromosomes to microtubules to allow chromosome segregation. What got you interested in studying that complex?
Yeah, so when I was a grad student, I guess I'd always found the cell cycle and cell division in general to be interesting, but I had a graduate project where I was going to work on a microtubule-associated protein. So I joined Georgiana Barnes and Dave Drubin's lab in 1997. And at that point, honestly, I don't think I'd actually even heard of what the Kinetic was as a thing. And I had a project where I was going to work on this microtubule-associated protein in budding yeast. And about a year or two into that, I started to realize that this protein was probably doing something else. This was a protein called DAM1. Mm-hmm. And I realized that in addition to holding onto a microtubule, it was at this structure called the Kinetic. Okay? And I think at that point, I kind of just fell in love with that structure. I think it was everything I wanted to try to understand about a cell. You know, it's a very physical structure. There's a lot of proteins there, as you mentioned. It's a very mechanical structure. It has to do stuff. There's sort of a business end of what it has to achieve. And it's a highly controlled structure. You need to coordinate that with the cell and the cell cycle and ensure that you're not generating errors. And I just really loved that sort of set of challenges, that you have this machine, you have to build it, mm-hmm. you have to physically do stuff, and you have to control it. And at that point, so when I started in 97, there was maybe 12 proteins that we knew about that were Kinetic mm-hmm. components. And so this was quite early days. Yeah. And so it was just beautiful to sort of see this expanding list of that molecular complexity and to really understand how this process works. So I fell in love with this DAM1 complex and the entire Kinetic and then have never really stopped.
Yeah, and so you said there were 12 proteins when you started. Mm-hmm. How many are there now?
So the number in human cells that I would say is about 110. And that varies in different organisms. So as much as I fell in love with this DAM1 complex, it is a protein that is this beautiful ring-like machine that holds onto a microtubule and carries a chromosome with it. And I loved studying that, but one of my advisors, Dave Drubin, pretty much every week would walk into the lab and say, okay, so have you found the human homolog of that yet? And I felt like I was really bad at blast searches for many years, but it turns out it actually doesn't exist. So there certainly are differences between different organisms as well. That some things are true in fungi and slightly different in human cells, so then I transitioned to our Shah Desai's lab where I focused on C. elegans kinetochores, which are holocentric. They occupy the entire chromosome, and they're very similar to yeast and very similar to human cells, and yet also different in other ways in terms of that machine. So probably the largest number of proteins we know is in human cells, and I think you can argue a little bit about what is a kinetochore component and what is a protein that is at that site. But I think as I would define it, about 110. And
so what makes something a kinetochore component as opposed to just in the vicinity?
Yeah, I guess the way I would define that is sort of the connectivity between the chromosomal DNA and a microtubule polymer, that you have to hold on to the nucleic acid, the chromosome. You have to be able to help mediate its segregation. You have to hold on to the microtubule. And so the things that are localized to the site that are playing a role in those interactions and holding that entire structure together, I would put in that category. But it's a really complex part of the DNA as well, right? And so there's going to be heterochromatin there and other specific things that participate in that way. And I think I probably wouldn't define those as kinetochore components. Yeah.
Okay, so you haven't, it's just the first day of the meeting, and so you haven't given your talk yet. So maybe you can give me a little bit of a preview about what you're going to present.
I mean, it's actually, in some ways, we already sort of touched on this, right? So I started grad school in 97, that's 20 years ago, and it feels a little bit to me like that was just yesterday, but it's been kind of fun for me to watch the field progress since then. And I think that some of the work that we've been doing lately has brought me back to those initial days as well, right? So, I love what this molecular machine is and the sort of complexity of how you build this and assemble it and drive things. But I also loved when I was working in budding yeast where you could do this sort of clean and clear genetic experiment. Okay, and so subsequently I transitioned to C. elegans, and then about halfway through in our shots lab started working on human cells. And so our lab is 95% we work in human cell culture. Okay. Human cells are gorgeous. They're beautiful, they're big. You can really nicely see the spindle and the kinetic core structure. But it was really hard to do a yeast-like experiment there. And I've always kind of been jealous still about that clarity and clearness by which you can create, a replacement or really ask what a protein is doing. And as many people now, I think the CRISPR-based approaches are really revolutionizing how we can think about doing that sort of experiment. And we still cannot back cross human cells and, do some of the beautiful kind of yeast genetics that you'd want. But I think a lot of the things are accessible there in a way that were not like even a year or two ago, right? And so we've been exploring that approach, and I think it's actually caused us to reimagine a lot of our expectations, okay? And so, for example, we take that 110 proteins. The kinetic core is really critical for what it means to be a cell in life, right? You have to, every time you make 2 cells, you have to distribute your chromosomes. You have to do that process correctly. You have to make sure that you not only physically achieve that, but you do that with high fidelity. And so that should be an essential process, right? And so for a cell to continue to survive and duplicate, the kinetic core should be essential. And yet in budding yeast of, say, 65 yeast kinetic core proteins, there's probably about 25 or 30 that you can eliminate from a yeast cell, and that yeast cell will grow apparently fine, okay? We know of a lot of human kinetic core proteins. I think actually it would have been very hard to say which of those are actually essential, right? So they're important, but are they actually, can you get rid of them, and can the cell still survive? And RNAi was a wonderful way to be able to do functional experiments. But that really simple thing, is this thing essential or is it not essential, is not something that we could access. And that's really been transformed by the ability to go in and clearly and carefully knock stuff out with CRISPR. And so we've been exploring that strategy to go through things, and a lot of those things I think have conformed to our expectations from diversity of other approaches and other organisms. A lot of those things have been a surprise. And I've liked those as well, right? And that simple metric, like, is this essential, really changes the way that you see a process, right? So one of the things that I will talk about, there's a signaling pathway called the spindle assembly checkpoint. And this is a pathway that senses when there is a problem in chromosome attachment and just holds up the cell until you get things right, and then you can progress and actually segregate and distribute your chromosomes. And in yeast, that's a non-essential pathway. So those proteins were first identified in budding yeast, beautiful work from Andrew Murray's lab and Andy Hoyt's lab. And they could make null mutants in those, and the yeast were fine, but they behaved like they could no longer hold up mitosis when there was a problem, okay? And, if you were to look in probably most papers that are out there, the simplistic statement would be, well, these proteins are non-essential in yeast, but they're essential in human cells. They're essential in vertebrate cells. So the checkpoint is an essential pathway in human cells. And I don't think that's true. And so it's true that many of those proteins, if you eliminate them, are essential. But you can create a cell that is genetically checkpoint defective, and it will grow just fine in culture, okay? And I think that actually makes you think very differently about what that pathway is and what its role is, and it meets much more the classical definition of what a checkpoint is, where it's a pathway that gets activated when there's a problem, but that you don't necessarily need it under all circumstances. So many of those proteins are critical for additional roles, but I think the checkpoint itself is not essential.
So what was the protein that was the biggest surprise for you? You thought this has to be essential, and it's not.
Yeah, and I think it's about context too, right? Yeah, I mean, in that case, probably these checkpoint proteins. So the ones that we weren't able to eliminate completely were, when I say we, the person who did this work in our lab, and I think that's also incredibly important, was Kara McKinley, who was a grad student in our lab. But so Kara made a collection of knockouts where she targeted more than 220 different genes that play roles in cell division. More than, I mean, these were things we would assume would be critical. Maybe 40% of them didn't show a strong and potent phenotype in cell culture, though, and we're quite confident that we're getting rid of these things. And so this complex that we can completely eliminate, which I really thought was going to be central and important, is this rod ZW10-Zwilch complex. Okay, and so those proteins you can get rid of, and the cell is fine. But there's been a lot of other things like that. And I guess it's a question of whether is it a surprise, right? So at the time, I'm like, okay, something's wrong. We should be getting, there should be something happening here. We should see these cells should be dying. But then maybe in retrospect, you start to think about it, and it's like, well, what was the data that made us think that it would be important or essential? I think that's not only been true with knockouts, but also with specific mutations. I think that there's a lot of things that can compromise a process without killing the cell. The cells are actually pretty robust, and they can tolerate things in cell culture, right? And so some of this sort of low rates of chromosome segregation maybe in an organism would be problematic.
Okay. And do you see a pattern? I mean, are things that are more sort of structurally integral show, are those more essential? More essential than things that are either more peripheral or may have catalytic I mean, can you categorize your hits that way? So I continue to be enamored with the Kinetic core, but I think one of the features of it that is fascinating is how flexible and plastic it is as a molecular machine. Okay, and do you mean that literally, like in terms of actually, deformability, or do you mean in terms of what can be accommodated in a functional unit? Literally it is. So it is a structure, but I mean figuratively. So you're right, literally it is a structure that can be highly deformed under force and tension, and is flexible in that way to be able to resist and handle that force. I mean figuratively in the sense that it is dramatically rewired during evolution. During the cell cycle, as you mentioned, it's a dynamic structure, it's assembling and disassembling, and so I think actually those answers vary between one organism and the next, and so we can. I think it's very important to define in human cells and in human cell culture who is essential for cell division, who is essential for chromosome segregation. But I think the answer is going to be very different from what that is in budding yeast, or what that is in maybe even in mice cells, right? And so we have had a protein that we've worked on that everything that we do to it in human cells, it seems really central and critical. And when we realized that there was a mouse knockout of that, the mice seemed to grow fine, that really surprised us. And I think I don't think it's so much about critical catalytic domain or structural role. It's about the way that the human kinetic core has chosen to do that. And so there are structural proteins that are non-essential in human cells, and those same proteins are structural proteins that are essential in budding yeast. And I love that rewiring. It really makes you think about, you take something like the ribosome or the proteasome, and these things are largely invariant, across eukaryotes, and the ribosome even pretty similar when you think about the way that bacteria do that. And here's a structure that just even over short evolutionary distances is changing and adjusting in different ways. That's very cool. So I have a question, sort of changing tack a bit. So it sounds like you've done this essentiality screen that's opening up a lot. What do you feel is like kind of beyond your reach? Like, what's the thing you want to know but you can't quite get there yet?
Yeah, that's great. And I mean, I think that's what keeps you going too, right? And I think maybe stepping back even further, right? So we talked at the beginning about how when I began, we didn't even have the molecular players, right? And so I think that there was the historic phase of the field when, from Fleming through to, say, the 60s, was very visual, like understanding what the, cell division was as a structure and actually being able to see the kinetic core early. And, I think that the modern molecular age of, say, the kinetic core and cell division began about 1987 when Bill Earnshaw, used the CREST autoimmune serum to find some of the first human kinetic core proteins, and there was very similar and comparable work in other model organisms, right? And 97, we still only had a dozen. The number exploded over the next decade. And I think that we've largely kind of hit that point where we have done the molecular cataloging, where we know who those parts are. And then that opens this new door, which is, okay, we have all these players, what are they doing? There's all these activities you need to achieve. How do they work individually and in combination to make that happen? And I think that for this meeting and for a lot of the work that we care about, that really is the focus, is bringing these together, not thinking about them in isolation, not thinking I found this new protein, but you have a machine that is an integrated molecular machine. How do you achieve that? Okay. And so I'm really enjoying the stuff that we're playing with for that. I think the thing that I would also really love to do moving forward is to think about how you do modulate what this structure is and how it works in an organismal context. And so I've been in some ways kind of careful about saying in cell culture. And I love human cell culture and I love our HeLa cells, and they're a very powerful place to explore things. But I think what's even more beautiful is to think about the 30 trillion cells in our body, and the diversity of tissues and organs and circumstances and developmental situations, and how you take these core cell biological processes and think about them in an organism. I'm really excited to be able to tackle that. And I think that some of that is starting to be accessible, but I think that there still needs to be some better tools to do that as well.
And so if there's one tool that you could just have to let you ask some new questions, what would it be?
Yeah.
Or what would it let you do?
Yeah. And I think mice to have a 24-hour doubling time or something, I don't know. Maybe easier ability to access those tissues and control things genetically in mice, and I think there's people who are excellent at that, and I think our lab needs to learn how to do those. I think that the organoid systems have opened those doors. I guess the tools are there, and they've been developed by diverse labs, and I think that there's something that we as a field need to do a better job of taking advantage of and harnessing. And so what is the missing reagent? I don't know that there is one. I think it's possible to make those. It's just now it's time to do that.
And so if you had all your resources at hand, you're starting your lab today, brand new, would you ask the same question, or is there something different that you would go after?
Yeah. I continue to feel, so I teach undergrad cell biology at MIT, and I also I love new people coming by our lab, and it's really fun for me to sort of talk about these processes and these ideas, and I'm surrounded by this unbelievably smart group of people, and most of the questions they ask, my answer is I don't know, and I love that, and I feel as much as we have been able to visually see this structure for 100 years. That these just basic things of what it means to be a kinetic core and what it means to segregate chromosomes and divide a cell, which is such a central biological process, there's things that we just simply don't know. And I think, so I'm so enamored by those ideas and questions that, I feel it's sort of like this addiction where, of course I would choose to do these again. I think coming back maybe to the first thing I said, I love the physical nature of biology. I love structures. And then I also love regulatory circuits. And the cell cycle is a nice example of that. I particularly like the interplay between those two. And I think that's why I continue to love the kinetic core, because it's a complex and beautiful example of that. You have to achieve this very physical thing, but it needs to be intricately controlled and regulated. And I think that if it were to be all solved tomorrow, which it's not going to be, I think that there would be other cell biological structures that I would also find fascinating.
And do you think, so given sort of the stages that the Kinetic core has gone through in terms of understanding it, are there lessons there for other large macromolecular dynamic complexes?
Yeah, and actually, I think in some ways we're behind some of these, right? So I really love reading, about the proteasome and the ribosome, or, I think the excellent work to reconstitute DNA replication in vitro, we've known about DNA replication for a long time, and many of those factors were identified earlier than kinetic core components, and there's still basic questions about them, and I think it did take reconstituting those processes to really understand that, and I think I look at those fields with envy, but also to try to learn how can we apply those lessons to what this structure is. But similarly with, say, DNA replication, and so I have two excellent DNA replication colleagues in my department, Steve Bell, who has reconstituted many of these things, and Terry Orweaver, who looks at these in a developmental context and trying to understand how different cells and tissues alter the way they're replicating things to create polyploidy or other kinds of amplifications. And so I think it's nice to look at those other processes and try to learn from them. I think that there's a lot of parallels for the cilia and the centriole, and there's nuclear pore and many other beautiful machines in the cell.
All right, well, I think we'll stop there.
Excellent.
Thank you very much.
It was great to talk to you.